The overall goal of this procedure is to identify somatic mutations in cancer-associated genes of cells from tumor tissue by using barcoded multiplexed DNA libraries followed by massively parallel sequencing. This is accomplished by first ligating barcoded sequencing adapters to DNA fragments isolated from preserved tumors. The second step of the procedure is hybridization to custom biotinylated oligonucleotides complementary to all the protein-coding exons of 279 oncogenes and tumor suppressor genes. The third step is to perform massively parallel sequencing of the barcoded pools from the captured libraries. The final step is to search the sequence data for cancer-associated alterations. For the 279 targeted genes, this procedure can reveal sequence mutations, small insertions, small deletions, copy number alterations, and select structural alterations.
So the main advantages of this technique over existing methods are that it allows one to investigate entire exons for both the common and rare mutations, as well as identify additional genomic alterations such as copy number losses and gains, and attain greater sensitivity in heterogeneous samples.
This method can help answer key questions in the cancer genomics field, such as what are the key driver mutations in various cancer types, and do these mutations in clinical specimens correlate with outcomes or response to targeted therapies?
The implications of this technique extends toward diagnosis and treatment of cancer because mutations may be identified prospectively and be used to help guide patients to appropriate therapies and clinical trials.
Visual demonstration of this method is critical.